Hello, students, and welcome to Science 30 from Rock the Diploma. I'm your teacher, Mr. Ferguson, and this journey will be a fun one as we move through the course. We're going to be starting in biology, moving into chemistry, and of course, we have the physics unit, and finally the last unit with energy, which is kind of a continuation of the physics unit, but also has so much other depth involved in it as well, connecting all of the units that we study here in Science 30. So let's begin by talking about the outline of the course. First of all, Science 30 is a very interesting course, how it's set up. The traditional route for students coming to Science 30 would be through Science 10, Science 20, and Science 30. This program was designed as a general science course for students to just get the basic outline of all the fields of science, and that was the traditional route. However, Science 30 has evolved over the years, and one thing that we've started to see within Science 30 is because post-secondary institutions are now accepting Science 30 for many of their programs, we see students coming from all different areas coming into Science 30. So that might be students who took Science 10, did Chemistry 20, then Chemistry 30. That might be students who did Physics 20, Physics 30, now are taking Science 30. We also have students who are taking Bio 20 to Bio 30 to Science 30. And so this creates a bit of a dilemma for us as teachers. We have some students that took the traditional route of Science 10, 20, 30, that would be the orange boxes. But to get into Science 30, all you need is a 20-level science course. So some students maybe just took Bio 20, and now here they are in Science 30. We could do this for all the other fields. Someone maybe took Physics 20 and then came to Science 30. Some go all the way to Physics 30, Chem 30, Bio 30. But one of the complexities is we have students with a wide range of backgrounds who are here taking the Science 30 Diploma Prep course. Some students maybe just took Science 10, then Chem 20, and then went straight to Science 30. So for that student who went straight from Chem 20 to Science 30, boy, oh boy, that student hasn't taken any physics or biology since Science 10. So a student like that, boy, you know what? The physics unit and the bio unit might be really complex. But then we have some students who have literally taken Physics 30, Chem 30, and Bio 30, and now they're taking Science 30 for a variety of different reasons. Now, who knows? Maybe it's for scholarships, maybe it's to just get a higher average to be able to then apply to post-secondary. Regardless, we have some students who've maybe not taken physics since their year in grade 10, and then we have some students who've taken Physics 30, Chem 30, and Bio 30. So talk about a wide range of students coming into this course. And then, of course, we have students like any diploma prep. Some are looking to get 100%. They're looking to get a great mark in the diploma for scholarship reasons, for post-secondary entrance. And some students, you know what, you're just hoping to get that 50%. And you're using this diploma prep to get you that extra push to succeed on the diploma and pass the course. And so once again, that's a huge range of students and their goals and expectations. And then finally, the other thing that I found that's very difficult teaching Science 30 is based off of the teachers that you've had within your school year. Now, this isn't to critique your school teachers. This is just to highlight one of the concerns that sometimes pops up in Science 30. So if we look at our three teachers at the bottom here, a bio teacher, a chemistry teacher, and a physics teacher, teachers who come in to teach Science 30, typically they have an expertise in one of the fields. So when they went to school, they either majored in physics, majored in chemistry, or major in biology. Now, this is where their passions are. This is where their expertise are. And so sometimes teachers, as we are all humans, sometimes we love teaching the stuff that we're most passionate about. So often I find students who come into Science 30 and they have had a teacher who you can tell has a passion for chemistry, has a chemistry background. So they've spent a lot of time doing chemistry and maybe potentially over-taught some of the concepts. Now, over-teaching a concept's not a bad thing. It challenges you and pushes you. But what I'm bringing up, though, is I'm going to focus purely on what you need to be successful in the diploma for Science 30. So I might actually not be teaching some of the stuff that your teachers taught you in your class, and that's not because they did anything wrong. They maybe over-taught a concept because they're passionate about it, because that's where their expertise is. However, my job is to get you ready for that Science 30 diploma. So there has to be a little bit of trust amongst us to know that I'm focusing on the program of studies and exactly what you need to have to be successful in the diploma. Now, let's look at the packages that you have provided to you. Now, you have package number 1, which is a very important package because that is going to be the package that's going to let you have your Science 30 data booklet. Okay, so we're going to refer to that data booklet, and it's going to allow you to just highlight those key points that are going to help you be successful. There's also the answer keys to all of the questions. Now, I do encourage you not to look at the answer keys. It's really easy for students to do a question, look at the back, look at the answer and go, oh, yep, that's what I would have chosen. But we know that's not always the case. So the answer keys are there after you've done the work, and then you can refer back to them to see how you've done. So I do ask you to refrain from looking at the answer keys until you've at least attempted every question on your own. And then, of course, they're there to let you know how you're doing. The Science 30 data booklet is of importance, and there's other important information in package 1, but outside of going to highlight the data booklet, we won't be looking at package number 1 as a group here during the course. Now, package number 3, this is another important booklet for you to look over. It has a practice diploma for you to try. It also has a bunch of sample questions. These are additional questions that we won't be covering within this online portion of the course, but you will be asked to take a look at them and use them as additional information to give yourselves a better understanding of what the expectations are for the questions and the difficulty of questions on your Science 30 diploma. So the checklist, the sample questions, and the practice diploma from package 3 is for your additional practice, and I highly encourage you to do all questions in that booklet to best prepare yourself for the diploma. So what are we doing here on the online portion of this course? We are doing package number 2. My goal, my job is to do every question in package number 2, as well as going over the notes and highlighting some of the key parts to allow you to be successful in those questions. So during this time, I will be teaching, but I also will be asking you to pause your screen at certain times to practice questions on your own. And then once you unpause it, I will go through those questions with little additional information to help you succeed. So with all that being said, let's begin the course with the very first unit, which is Unit A, Biology. And so this course, there's 2 portions to the biology unit. There is the heart and how it works. Now, what goes in the heart? Well, that's blood. So we study blood. And then specifically, one portion of the blood in the white blood cells, we further discuss immunity. So that's the first section. And the second section is on genetics, and we will continue with that. But let's first tackle the heart, blood, circulation, and immunity. I now ask that you pause your screen to do the next 4 questions on your own. When you've tried all 4 questions, I ask that you unpause, and we will go through those questions together, and hopefully you find some great success. So I ask that you pause your screen now. All right, welcome back from doing those 4 questions. Let's take a look at them. Of course, you could have went to the answer in the back, but the whole point of going through this online course is going through together with the video and with me as your instructor. So hopefully you haven't had the chance to go and peek, and that you're able to follow along and listen to what we are doing here. So in this question, they are asking us that during chemotherapy, a drug is injected into a vein in the patient's arm. The sequence in which the drug moves through the 4 parts of the circulatory system listed above is, now here's the thing, it first went into a vein. And remember, veins bring deoxygenated blood back to the heart. Outside of the pulmonary vein, which brings oxygenated blood to the heart, all veins are carrying deoxygenated blood with the exception of that pulmonary vein. So when we look at the way blood would come back into the heart, we know that it comes in first through the vena cava, either superior or inferior, so the top or the bottom, but it comes in. Now, from the perspective of the patient, we are on the right side of the heart, and it'll go from atrium to ventricle. And then from ventricle, that less muscular side of the heart is going to pump the blood away from the heart. So that's an artery, but it's going to be deoxygenated blood. And usually arteries carry oxygenated blood, but because it is finally the pulmonary artery, that blood is going to get pumped away from the heart, going towards the lungs to become oxygenated. 1, 2, 3, 4. It doesn't get more simple than that. Now, question number 2 and 3 require us to look at this diagram of an artificial heart. Now, this is important to note that maybe you've never seen a heart like this, or maybe your teacher never talked about a component like this. And this is a good time to introduce the fact that in Science 30, and actually all science courses within Alberta Education, we have 3 different things that we try to teach in terms of outcomes. There is what we call the knowledge outcomes, which we just symbolize with a K. There is the skill outcomes, which we just show as an S. And then there are the STS outcomes, which stands for science, technology, and society. Now, the STS outcomes, this would be a technology example of looking at this heart. So what we as teachers do is we can't highlight every type of societal issue or technological advancement, but what we try to do is get students to start to analyze these types of questions so that they can be successful at synthesizing information and being able to read a new diagram that they've never seen and be successful in breaking it down. So this is going to require some knowledge, these questions, as well as some comfortability with understanding technology and science and how it works within our society. Now, just as a final note, the skill outcomes, those would be things like graphing, doing different experiments, knowing manipulated, responding, and controlled variables, and these are all very important things. So, but in terms of this question, we are going to be looking at some knowledge as well as some STS outcomes. So one type of artificial heart contains rotating components that help push a constant stream of blood through 2 separate compartments. As a result, a patient with this type of artificial heart will not have a pulse or a heartbeat. Now, the diagram of blood is shown to us here. So we see that deoxygenated blood enters in through 1. Then that deoxygenated blood looks like it's leaving 3. Then it comes back as oxygenated blood in 4. And then that oxygenated blood leaves the heart at number 2. So let's take a look at the question. It says, which of the following structures prevents the backflow of blood within the normal human heart? Now, here's the deal, you guys. Now we're talking about just a normal human heart. So we are moving away from the 2-compartment artificial heart, but it's just a question. So what prevents backflow of blood within a normal human heart? That would be just the valve that we talked about, not specific names of the valves, just valves. That's what we need in Science 30. Now, question number 3 says match each structure numbered in the diagram of the artificial heart and the diagram above with the description below. Use each number only once. Okay, so which one sends blood to the brain? Well, you know what? We probably want to have oxygenated blood coming to the brain. So that would be component number 2. Let's go back here. We can look back at the diagram. We can see that here it is, oxygenated blood leaving that heart. That's what we want to have coming to our brain. Oxygenated blood comes to the brain, sends blood to the lung. That would be component number 3. That would be sending deoxygenated blood to the lungs to become oxygenated. Well, once again, number 1 would be deoxygenated blood coming into the heart. So that would have to be attached to our vena cava. The vena cava is bringing deoxygenated blood to the heart to get pumped through. And finally, the last one, attached to the pulmonary veins. The pulmonary veins are the ones that are bringing in oxygenated blood. That's the exception, right? Because normally veins are bringing deoxygenated blood, but component number 4 is bringing in oxygenated blood. If we move back in the slide, you can see oxygenated blood is coming into the heart, and that would only happen within a natural, normal human heart through the pulmonary veins. 2314 is your answer to that question. A common heart defect occurs when a hole between the left and the right side of the heart, remember that's the septum, right? The septum is what separates the right and the left-hand side of the heart. And this hole results in an abnormal flow between chambers as shown in the diagram to the right. So just for question number 4, we ask ourselves, which of the following rows identifies the affected heart chambers in the diagram above and describes a result of the heart defect? So you can see in the diagram that we are looking at the hole happening in the upper part of the heart where we would find our atrium. So the atria are where the affected heart chamber is happening. That's where the hole is. And what would be the result of this? Well, as you can see, if you eliminated C and D, as I hope many of you would, knowing that the chambers or the atria that are being affected, you have a choice of, does it lower the heart rate? Well, maybe it potentially could, and I know students don't like hearing best possible answer. Maybe it could. I don't know, but I do know that the type of blood that are found in the two atria are oxygenated and deoxygenated. So that hole would then obviously allow the mixing, and that becomes our best answer. So one thing that we see though is heart rate will increase because the heart is responding to that mixing of the oxygenated and deoxygenated blood. So it's not going to be A, but regardless of whether you knew it or didn't know it, whether the heart rate would increase or decrease, we're looking for the best answer. And B provides us a 100% sound answer that all students in Science 30 would be able to say, there is a correct answer, and B is your answer. All right. So now that we've completed those first 4 questions and we've got our kind of toes in the pool a little bit, we got to dive straight in. So as we continue our journey within the circulatory system, of course, the heart is really important. But once the blood leaves the heart, we now have to discuss and look at where does the blood go? And it leaves our heart through arteries, comes back to our hearts through veins. But there's 3 other important aspects that we have to look at in terms of blood vessels when blood is being transported throughout the circulatory system. So from arteries, these are very thick, elastic muscle layer. These carry blood away from the heart. And then they go into smaller branches of arteries called arterioles, and these connect arteries to capillaries. Now, capillaries are where we have the site of gas exchange. This is a site of nutrient exchange, as well as waste products from the blood that come from muscle tissue. This is the site of exchange where, once again, we see exchange of oxygen to the muscles. The muscles in return give carbon dioxide. This is also a spot where we give nutrients and take waste such as lactic acid away from those muscles. And so the capillaries are kind of the spot that connects to our muscles. And then after that, the capillaries then bring the blood back through venules which those venules then form veins, and veins once again return the blood back to the heart. Now, guys, you have to understand the structure. You have to understand what they look like, what their function is, which we've discussed in this brief introduction, but we also have to know general pressures. Now, you don't need to know specific numbers, so you don't have to memorize all of these numbers in the third column. But in this third column, please understand the nature in which not only does the blood pressure occur happening from highest blood pressure to low blood pressure, but also understanding how that structure or that pressure explains maybe what they look like. So of course, arteries have the highest pressure, taking blood away from the heart. That makes sense that they have a thick elastic muscle layer. With all that pressure, you would need to have a thick elastic muscle layer. Now, as we move away and the pressure gets lower and lower and lower, we can see that the actual structure of these blood vessels also gets well, it explains why with veins it's a thin elastic muscle layer. Well, it's a thin elastic muscle layer because that's where we see the lowest pressure. So please be familiar with this chart. Take this opportunity to read it and digest all that information. Very important to know these different components of blood vessels and their structure, their function, and the blood flow regarding pressure within each of those blood vessels. So here's a great visual as well. Some of us we like reading and digesting information that way, and some of us are visual learners. So as we can see here, the artery has a thicker wall, where veins have a thin layer of muscle. Capillaries, they are very small, great surface area, they're small and creating lots of surface area because that's where all the exchange of gas, nutrients, and waste occurs.